Good morning, students. Today let us study Bohr effect and cooperativity. Okay. Now what is Bohr effect? In vertebrates, dioxygen enters into the blood in the lungs or gills. You know that in vertebrates the dioxygen is nothing but oxygen enters into blood, from where it enters, either lungs or gills. Now what happens to the partial pressure? Partial pressure of dioxygen is relatively high, and what is that order? 10.3 into 10 to the power 4 pascals or 100 millimeter of mercury. Okay. Under ideal conditions in the lungs with mixture of inhaled and unexhaled gases. You know that in the lungs inhaled gases are present as well as unexhaled gases are also present, and because of this in the lungs partial pressure will be relatively high. And what is the order? This is the order. Okay. 10.3 into 10 to the power 4 pascals. Alright. Now it is then carried by the RBCs, that is red blood corpuscles, to the tissues. Okay. So this inhaled dioxygen or oxygen is carried by RBCs to the tissues, where partial pressure of oxygen is considerably low. Now in the tissues partial pressure becomes low, and what is that order? 2.5 into 10 to the power 3 pascals to 5 into 10 to the power 3 pascals. Okay, or that is also equal to 20 into, that means 20 to 50 millimeter of mercury. Okay. Next slide. Now this is the equation taking place, that means this is the reaction taking place in the lungs or gills. Hb plus 4 oxygen, this is nothing but dioxygen. So hemoglobin combines with the dioxygen. Okay, 4 molecules of dioxygen, where? In the lungs. Alright. And what is the compound formed? Hemoglobin is tetradioxygen. So HbO2 4 times. Okay. Now in the tissues what happens? This tetradioxygen, that HbO2 4 times, combines with 4 myoglobin molecules. Okay, giving 4 MbO2. Mb is nothing but myoglobin and Hb is nothing but hemoglobin. Okay. So MbO2 plus Hb plus hemoglobin, that means this bond will break, hemoglobin and oxygen, this bond will break. Okay. So that you get what? Mb and then O2 4 times, this is 4 times, this is 4 times, so you get 4 times MbO2. Okay. And then this bond will break, Hb and dioxygen, therefore you get Hb. Okay, plus Hb. Now hemoglobin has an ambivalent function. Now what is the ambivalent function of hemoglobin? It should bind to dioxygen firmly. Okay. One is to bind with the dioxygen firmly and carry it as much as possible to the tissues. Okay. So it will bind with the dioxygen firmly and then carry it to the tissues as much as possible. Alright. Now what happens when it reaches there? So as soon it reaches there, it should readily release it to myoglobin. Okay. So hemoglobin combines with dioxygen and then reaches to myoglobin. As soon as it reaches to myoglobin, it will release that one oxygen, okay, which can store it for oxidation of glucose. Now why oxygen is required? For the oxidation of glucose. Okay, glucose is present, you know that. So it will combine, or oxidation of glucose will take place to give what? Carbon dioxide and water. Okay. Next slide. Graph between percentage saturation and partial pressure of oxygen. This is the graph which gives the percentage of saturation, okay, and then partial pressure of oxygen. So partial pressure of oxygen is given in units torr. Torr is the unit to express what? Partial pressure of oxygen. Okay. So this is the graph. So this graph is obtained for hemoglobin, and dotted curve, this is the dotted line. Okay, this is the dotted curve obtained from myoglobin. Okay, this is for myoglobin and then this is for hemoglobin. Okay, and you can see here myoglobin curve lies above what? Hemoglobin. This is below. Okay, hemoglobin curve will be below and myoglobin will be higher level. Okay, you can see here. As the partial pressure of oxygen increases in the case of myoglobin, okay, percentage saturation increases, there is a sharp increase like this, and then Finally, at high partial pressure region it remains constant. You can see here constant. Okay, so this is the high pressure partial pressure region 80 to 100. So at this partial pressure it will what becomes constant. Okay, and where it will take place in lungs. Okay, it will remain constant in lungs. Okay, high at a high pressure region. Okay, so this is the lungs partial pressure. Now in the muscle, this is the muscle region. Okay, muscle region the partial pressure is low. You can see here. Okay, 20, 40 up to 40 maximum. Okay, so this is taking place at muscles. So at muscles, myoglobin partial pressure okay reaches here percent saturation rapidly here like this. Okay, whereas in hemoglobin less rapidly. Okay, slow there is an increase. Can you see here? Okay, slow there is an increase like this, and then there is rapid increase. Okay, so this is the graph obtained for myoglobin, hemoglobin, percent saturation versus what? Partial pressure. Alright, next slide. Hemoglobin and myoglobin has almost similar binding effect for oxygen at high partial pressure of oxygen. You have seen in the previous graph, at high partial pressure region, okay, you can see constant, correct. So, or in other words, similar binding effect. The curve remains constant, that means similar binding effect, okay, with the oxygen. Now, hemoglobin is poor oxygen binder at lower pressure of oxygen. This also you have seen in the previous graph, correct. So, it will slowly increase, okay, slow increase, okay, in the low pressure region. That means it has got poor binding effect in the low pressure region, okay. Then, myoglobin has higher binding effect at lower partial pressure of oxygen, whereas in the case of myoglobin, it has got high binding effect or higher binding effect at lower pressure region, because it has got sharp increase at lower pressure region. Okay, what about hemoglobin? This I already explained you. In the case of hemoglobin, there will be slow increase. Okay, high low pressure region, whereas in the case of myoglobin at the low pressure region there will be sharp increase or higher binding effect. This shows that myoglobin has higher binding effect at lower partial pressure region, okay, whereas hemoglobin has a low or poor binding effect, okay, at a low pressure region. Alright. Now HbO2 when reaches muscles lower partial pressure oxygenation, when hemoglobin combines with the oxygen and reaches the muscles, the partial pressure decreases. Okay, next slide. Tissue needs more oxygen. Tissue needs more oxygen to continuously utilize and convert into carbon dioxide. Why tissue needs more oxygen? You know that tissue's glucose is present. So, oxidation of glucose taking place. So, glucose, what is the molecular formula? C6H12O6. This equation is not given here. So, C6H12O6 plus oxygen will give you carbon dioxide and water. This is the reaction. Okay. So, you get one carbon dioxide water, that equation is not given. So, this carbon dioxide water is obtained from the oxidation of glucose. Okay. Now, this carbon dioxide will combine, okay, with the water or hydrolysis taking place to give what? Bicarbonate ion and hydronium ion, okay, H3O plus. Now, this bicarbonate ion is acidic in nature, okay, this is an acidic in nature, therefore, pH becomes low or it will give lower pH value. So, lower pH favors the release of oxygen from hemoglobin to myoglobin. Since the here acidic radical is formed or acidic component is formed, pH becomes low, okay, therefore, at that acidic pH value, okay, hemoglobin will give oxygen to one myoglobin. Okay, hemoglobin first carries during respiration. You know that oxygen will reach the lungs. So, it will combine with the hemoglobin. Hemoglobin will be carrying one oxygen. Then when it reaches to myoglobin, because of this acidic condition, okay, low pH, it will give it to myoglobin. Okay. So, oxygen affinity of hemoglobin varies with pH. You know that oxygen affinity of hemoglobin varies with pH. This variation of oxygen affinity of hemoglobin with the pH of the medium is called Bohr effect. Okay, what is Bohr effect? The oxygen affinity of hemoglobin varies with one pH of the medium. Okay, so it depends upon the pH of the medium. This effect is called by one Bohr effect. Okay, next slide. Now this is the symbolic representation of Bohr effect. Next slide. Cooperativity. Now let us study what is cooperativity. You know that iron is present at the center of the heme. Okay, you have already learned the structures of heme, hemoglobin, myoglobin. Okay, you know that iron is present at the center of heme. Okay, Now it will be bonded to 4 nitrogen atom of 4 pyros of porphyrin ring. This also you know. Iron will be present at the center in heme and it will be surrounded by 4 pyral molecules of what? Porphyrin. Okay. And 1 nitrogen of imidazole side chain of histidine. Another nitrogen atom it will be bonded, that is 5th nitrogen. 5th nitrogen is nothing but the chain of histidine residue, okay, or histidine residue. That means imidazole side chain of histidine. In deoxyhemoglobin ligand field is weak. If you consider deoxy means without oxygen, okay, just HB, not HbO2. Deoxyhemoglobin ligand field is weak, okay. The complex has a high spin with configuration. Now, what is the configuration of ion in this field? T2G4 and then Eg2. You know that Eg there are 2 orbitals and T2G 3 orbitals are present. Okay, you know that in the coordination chemistry chapter you might have learned. Okay, there are d orbitals are splitting up into 2 levels. Okay, T2G and Eg levels. And Eg level there are 2 orbitals. Okay, and T2G level there are 3 orbitals. So T2G 4 electrons are present, 3 orbitals 4 electrons are present, okay, and Eg 2 orbitals 2 electrons are present, okay, and electrons are unpaired, okay, each orbital 2 orbitals 1 each, and T2G 4 means 3 orbitals 1 each and then 1 is paired. That means here also you have unpaired electrons, okay. Therefore, if there are unpaired electrons are present, that magnetic field we call it as a paramagnetic, or that complex we call it as paramagnetic complex, okay, correct. Next slide. Iron is substantially large and in low spin state of iron configuration T2G6 and Eg6. Suppose it is in low spin state, then the configuration will be T2G6 Eg0. That means Eg orbitals no electrons are present, vacant, okay. T2G there are 3 orbitals, each orbital contains 2 electrons, so 2 into 3 is 6. That means all are paired up, correct, no? So all are paired up, so T2G6. Because of repulsive effect of 1 of Vg electron, okay. So present in dx squared minus y squared orbital directed towards the 4 net porphyrin ring. So this shows that all electrons are paired up, that means it is diamagnetic in nature, okay. If it is unpaired, it is paramagnetic. So in that case, iron and nitrogen distance is larger than the size of the central hole in porphyrin ring, okay. If there is no oxygen, that means no oxygen is bonded, it will be one, it will not fit into that oxygen. This Fe distance will be larger than the size of the central hole. So deoxymyoglobin ion lies above the plane of 4 nitrogen atoms by 7, okay, 70 and a square pyramidal structure, okay. This you need not worry much. In the next slide you can see Oxy and Deoxy Hemoglobin. So this is the way of representing Oxy and Deoxy. This is one Oxy Hemoglobin, because oxygen is present. Can you see here 4 nitrogen atoms of the 4 pyral rings, okay, and this nitrogen is of histidine residue. This is histidine residue, okay, histidine residue, and this oxygen is bonded. Therefore 6 sites are there, okay, 1 2 3 4, 1 here nitrogen, 1 for oxygen, okay. What about here? This is the deoxy. This is deoxyhemoglobin, this is oxyhemoglobin, because oxygen is present, okay. This is deoxy because no oxygen. So here will be only 5 sites are here combined, that means placed. So 4 nitrogen, this nitrogen is missing here, and then this is what? Nitrogen, obviously no oxygen here, okay. This distance is 70 picometer, okay, here this is 200 picometer, okay. Next slide. Oxygen bonds to 6 coordination, okay. So in oxyhemoglobin, oxygen bonds to 6 coordination, all right, results in a low spin complex. So in that case, a low spin complex will be formed. So what is the configuration in the low spin complex? That in the low spin complex means all the electrons are paired up, that means T2G6 and Eg0, okay. So diamagnetic, so that complex is diamagnetic, all right. And iron and nitrogen distance is same and it is coplanar. In that case, Fe distance will be same, okay, and that compound is called coplanar. The release of strain energy of the square pyramidal complex compensates for the loss of spin pairing energy. So whatever strain energy will be released when it has got square planar complex, that will compensate for the loss of spin pairing energy. Now, therefore, the coordination of oxygen results in dropping of ion into the plane of porphyrin ligand. So, ion will be dropped into the plane of porphyrin ligand. Alright, next slide. HpO2 is low spin octahedral complex. Ion lies in the plane of porphyrin ring. The net results of the interaction of Hp is to increase the affinity of oxygen of the second heme site and so on. Now, hemoglobin, you know that it contains 4 heme groups and the cooperativity is observed. Now, what happens in the myoglobin? Myoglobin contains 1 heme group. Okay, this you know, since you have already learned the structure of myoglobin, it contains 1 heme group. Such cooperativity is not observed. In myoglobin, no cooperativity because it contains only 1 heme group, whereas the hemoglobin contains 4 heme, therefore, cooperativity is observed. But change of geometry after binding oxygen increases affinity for oxygen more than hemoglobin at low pressure of oxygen in the molecule. So, in the case of myoglobin, the binding with oxygen results in more of what affinity for oxygen at a lower pressure region, that is you have seen in the graph. Okay. Whereas in the case of hemoglobin, low pressure region binding effect will be less. Okay. Next slide. So, this is another way of representing. Okay. You can see here hemoglobin, this is a molecule protein, this is the protein chain, this is the porphyrin hole. Here ion will be slightly above because it is not fitting properly into the hole, this is the hole. Okay. So, ion will be up here. Now, what happens when oxygen links to the ion, that means oxyhemoglobin. So, this will pull this protein chain down. Okay. So, this is the protein chain, it is just like in the form of elastic chain. Okay. So, that will be pulled down. Okay. So, this will be pulled down. Okay. So, that this ion whatever present here ion up, that will fit into this hole. Okay. This is known as cooperativity. So, here there is no cooperation, here there will be cooperativity. Okay. It will fit into the hole. Okay. When there is oxygen binds to the ion molecule. Okay. So, as one ion binds an oxygen molecule, the molecular shape changes to make the binding of additional oxygen molecule easier. Okay. So, one ion binds an oxygen molecule, the molecular shape changes. So, this is the change in shape of the molecule, correct. Here there is one unusual molecule we can say. So, here shape has been changed. So, as ion binds an oxygen molecule, the molecular shape changes to make the binding of additional oxygen molecule easier. Okay. So, additional oxygen molecule binds easily. This phenomenon is called what? Cooperativity. Okay. So, with this chapter is over. Alright. So, go through all these slides. Okay. Any clarification, please contact me. Okay. So, suppose if offline classes starts, so when the offline classes starts, so I will redo it. Okay. Any difficulty, I will redo it. Okay. So, at that time we can have one discussion. All right. Thank you.